Hi viewers, in this class we are going to discuss stem cells, types and their applications. Under this topic we will see introduction to the stem cells, properties of stem cells, sources of stem cells, classification of stem cells and finally applications of the stem cells. Let us see the introductory part. Stem cells are the cells found in multicellular organisms. They are characterized by ability to renew themselves through mitotic cell division and differentiate into diverse range of specialized cells. Human body is made up of 200 different kinds of specialized cells. Some of these include muscle cell, nerve cells, fat cells, hepatic cells and skin cells, etc. All these cells in the body are derived from undifferentiated stem cells. So, stem cell is a cell that has the ability to continuously divide, differentiate and evolve into various kinds of cells and tissues. In developing embryo, stem cells can be differentiated into all specialized embryonic tissues. In adult organism, stem cells and progenitor cells act as the repair system for the body by replenishing specialized cells and also maintain normal turnover of generating organs. To ensure self-renewal, stem cells undergo two types of divisions. They are symmetric divisions and as well as asymmetric divisions. In symmetric division, stem cells give rise to two identical daughter cells. In asymmetric division, stem cells produce only one stem cell and a progenitor cell with limited self-renewable potential. Progenitors generally undergo cell division before differentiating into mature cells. And for the first time, THOMSON separated stem cells from the human embryo. Stem cells and progenitor cells act as the repair system for the body, replenishing the specialized cells. See here, this is the stem cell which can be modified into muscle cell, fat cell, bone cell, blood cell, nervous cell, epithelial cell, immune cell and sex cells. See here, symmetric cell division and asymmetric cell division. In the symmetric cell division, they give rise to two identical daughter cells. Here two identical cells, here two identical cells. Whereas in the asymmetrical cell division, two different cells. So one is a normal progenitor cell and another one is a stem cell. Two different cells. Here there is a hierarchy of the stem cells. This is a totipotent stem cell, pluripotent stem cells, and these pluripotent stem cells can capable to form different kinds of cells apart from the blood stem cells, RBC and WBC. Here other stem cells, they may produce muscle, nerve, bone and the other tissues. And coming to the properties of the stem cells, they are unspecialized cells. They are unspecialized cells. They show self-renewal, that is, stem cell can proliferate or They are capable of replenishing themselves for a long period of time by dividing. And there is a obligatory asymmetric replication ability where stem cells divide into one father cell that is identical to original stem cell and another daughter cell that is differentiated. And stochastic differentiation, when one stem cell develops into two differentiated daughter cells, another stem cell undergo mitosis and produce two stem cells identical to original. And another ability is potency to differentiate. That is, it specifies the differentiation potential of stem cells. They can differentiate into specialized cells such as nerve or heart cell. And coming to the plasticity, this is the property of stem cell for conversion of one tissue stem cell which can form another tissue cell. So here at a glance, unspecialized cells, they undergo self-renewal and obligatory asymmetric replication, stochastic differentiation, potency to differentiate, and the plasticity are the properties of stem cells. See here, the number 1, it shows the symmetrical cell division. Number 2, asymmetric cell division. Number 3, progenitor division. Number 4, terminal differentiation. And here, this is the stem cell, and this is the progenitor cell, and this is the differentiated cell. All of them they have shown in different colors. See here, there is a asymmetric replication where there are two cells are emerged from a single stem cell. One is stem cell, another one is a differentiated. And the stochastic differentiation, one stem cell develop into two differentiated daughter cells. Another stem cell undergo mitosis to produce two stem cells identical to original. And coming to the sources of stem cells. Stem cells originate from two main sources. One from adult body tissues and another from embryos. Embryonic stem cells are there, adult stem cells are there, and cord blood stem cells. Embryonic stem cells, adult stem cells, and the cord blood stem cells. This is the embryonic stem cells. They are extracted from the embryos and are thought to hold the most potential because these cells can give rise to virtually any specialized cell in the human body. See here, there is a oocyte and sperm. Sperm fertilizes the egg, and here the zygote, and this is the blastula, blastocyst, inner cell mass. So these are the embryonic stem cells. If it develops furthermore, it gives rise to the formation of the fetus, embryo. So the inner mass to be considered as the stem cells of the embryo, from which we can differentiate these embryonic stem cells either into blood cells or into neurons or into cardiac muscle and so on. Whereas the adult stem cells, these are present in the adult tissues such as bone marrow, brain and the blood, but are limited in potential relative to embryonic stem cells. When we compare these adult stem cells with the embryonic stem cells, the potency is very limited. They may be present in bone marrow, mesenchyma, pancreas, liver, muscle, neuron, skin, dermis, adipose tissue, placenta, and the cord blood. And coming to the cord blood stem cell. The sources of these stem cells are derived from the cord blood and has enormous potential in treating a disease. Stem cell can be harvested from umbilical cord after delivery with no harm to the baby. So people donate the cord blood and other store it. The harvesting of stem cells can be expensive, but the advantages for future needs include the stem cells are easily accessible, less chance of transplanted tissue being rejected if it comes from the recipient's own body. These are the benefits of the cord blood stem cells. See here, these are the hematopoietic stem cells which can differentiate it into RBC, WBC and blood platelets. Here mesenchymal stem cell, they differentiate into chondrocyte which gives rise to the formation of cartilage, osteoblast which gives us the formation of bone, fibroblast, adipocyte, neurons under the glial cells, skeletal muscles and the epithelial cells. And coming to the classification of stem cells, there are two factors. One is based on ability to differentiate and another one based on a source of stem cells. First one is the based on ability to differentiate. They are classified, stem cells are classified into totipotent, pluripotent, multipotent, unipotent and the oligopotent kind of stem cells. Totipotent stem cells are found in early embryos. Each cell can form a complete organism, whereas pluripotent stem cells exist in undifferentiated inner cell mass of blastocyst and can form over 200 different cell types found in body. Multipotent, these stem cells are derived from fetal tissue, cord blood and adult stem cells. Although their ability to differentiate is more limited than important stem cells. And coming to the unipotent, these stem cells can produce only one cell type of their own but have the property of self-renewable which distinguishes them from non-stem cells like the muscle cells. And finally the oligopotent, these stem cells can differentiate it into only a few cells such as lymphoid or myeloid stem cells. Another classification is based on the source of the stem cell. This is based on the source of the stem cells. They are embryonic stem cells, embryonic germ cells, adult stem cell, myeloid progenitor cells, lymphoid progenitor cell and umbilical cord stem cell. Umbilical cord stem cell. See here, based upon the potency, totipotent, pluripotent, multipotent and unipotent just now we have discussed. So the best examples for the totipotent which are present in the embryonic stem cell, pluripotent, the stem cells of the inner mass and these are the multipotent stem cells. Coming to the embryonic stem cells, these are inner cell mass of embryonic blastocyst. This can produce and differentiate into all types of cells of the body. When there is no stimulus, the cells divide, grow in number, and form pluripotent stem cells. These embryonic stem cells are useful for understanding of different kinds of diseases. Whereas embryonic germ cells, these are derived from the part of human embryo or fetus that will form gonads of embryo, ultimately produce eggs or sperm. These are formed in the embryo at 5 to 6 days old. Like adult cells, these cells are tissue specific and produce cells within particular tissue or organ in which they are found. Adult stem cells, these are undifferentiated cells found among specialized or differentiated cells in almost all tissues or organs of the adult body and appear to have a more restricted ability to produce different cell types and to self-renew. And these are also called as the vegetative stem cells, which are highly tissue specific. And these stem cells replace or regenerate the damaged or dead cells. Hence, these are also called as the vegetative stem cells. These stem cells are tissue specific and form new mature cells according to the organ or tissue they are present in. The best example, skin Langerhans cells of the skin, and satellite cells of the skeletal muscles, and pericytes of the smooth muscle. So in the smooth muscle, there are pericytes, intestinal stem cells, brain stem cells, and the mesenchymal stem cells, and bone marrow cells. These all are the best examples for the adult stem cells. The bone marrow stem cells are hematopoietic stem cells or hemopoietic stem cells. These are the best example for the adult stem cells, which are partly pluripotent and fully multipotent in nature. Each hemopoietic stem cell can divide to form two primary stem cells, out of which one cell is reserved as the mother stem cell and other divides to form two secondary stem cells called as the progenitor cells, or multipotent stem cells called myeloid progenitor cell and the lymphoid progenitor cell. Let us see myeloid progenitor stem cell. These cells produce various types of unipotent stem cells like erythroid progenitor, megakaryocyte progenitor, basophil progenitor, eosinophil progenitor, granulocyte monocyte progenitor, and finally the dendritic cell. These are the progenitors. Erythroid progenitor that produce the erythrocytes or RBC, megakaryocyte progenitor that produce the blood platelets, basophil progenitor basophil, eosinophil progenitor eosinophil, granulocyte monocyte progenitor that produce neutrophils and the monocytes, and finally the dendritic cells. Lymphoid, the myeloid progenitor, all these stem cells are differentiated into mature cells by forming different stages of cells. For example, erythroid progenitor cell is differentiated into proerythroblast, erythroblast, normal blast, and reticulocyte, and finally becomes the RBC. So there are several stages in their journey. Coming to the lymphoid progenitor cell. This cell produces unipotent stem cells, namely T cell progenitor, B cell progenitor, and natural killer cell. T cell progenitor cell migrates from bone marrow to thymus gland, where they get matured as the T lymphocytes, and B lymphocyte progenitor form B lymphocytes in the bone marrow itself. This is all about the lymphoid progenitor cell. See here, this is the multipotent hematopoietic stem cell, HSC. And now that modified into myeloid progenitor and lymphoid progenitor. Myeloid progenitor gives rise to the formation of megakaryocytes and to the platelets. Here erythrocytes, mast cell, myeloblast, they form into basophil, neutrophil, eosinophil and monocytes. The monocytes shows diapetesis and form the macrophage. The lymphoid progenitor that form the natural killer cells, and these small T lymphocytes they become T lymphocytes and B lymphocytes. B lymphocytes they get mature to produce the antibodies, that is the plasma cell. Here this is the self-renewing stem cell, early progenitor cells, and they differentiate into neuron, oligodendrocyte, and the astrocyte of the nervous system. Umbilical cord stem cells. Umbilical cord also contains stem cells. They are present in the form of a jelly known as Wharton's jelly. These are less invasive and show greater compatibility and less expensive. These exhibit the following three features. They are plasticity, homing, and the engraftment. Plasticity, it is the potential of stem cells to change into other cell types, even like the nervous cells, that is the plasticity. Homing, it is the ability of stem cells to travel to the site of tissue damage. It is a homing effect. And engraftment is the ability of the stem cells to unite with other tissues. That is the engraftment. So these are various examples of the stem cells. And coming to the applications of stem cells, there are different applications. They are used in various fields. Let us see one by one. Replace diseased or damaged cells, used to develop genetics. They can repair spinal cord and heart tissue, improve cardiac function in victims of heart failure, reduce pancreatic cancers and type 1 diabetes, heart infarction, baldness reversal, blindness reversal, spinal cord injury repair, Alzheimer's and Parkinson's disease, osteoarthritis, traumatic brain injury repair, replacing missing teeth, repair hearing, wound healing, and here all these are shown in a single triangle. The miracle uses of stem cells in your body. And therapies which are done by using the stem cells. Adult stem cell therapies are there, ASC, embryonic stem cell therapies, ESC, and induced pluripotent stem cell therapies, iPSC. There are different therapies done by utilizing the different kinds of stem cells. And the role of stem cells in the treatment of diabetes mellitus. Diabetes mellitus, it is a most common disease that is happened due to the lack of a hormone which is called as the insulin hormone. Basically, this is produced by the endocrine gland pancreas. The pancreas has the endocrine portion in the form of the follicle of Langerhans, islets of Langerhans, and there are different kinds of cells, alpha, beta, gamma, delta, and out of these beta cells they have the ability to produce insulin hormone. If there is a lack of insulin hormone, that leads to the diabetes mellitus. In this disease, blood sugar levels are increased and the excess sugar that will be excreted out through the urine. That will be excreted out through the urine. To regulate or control the blood sugar levels, the insulin is used under the supervision of the physician. And some research practices, favorable results have been obtained by transplantation of the pancreas. Nowadays, they are trying to transplant islets of Langerhans. Many researchers focused on embryonic stem cells. For example, they used the mice model. They implanted stem cells like human embryonic pancreatic cells or purified human Langerhans islets, etc., from different sources and studied the levels of insulin. They came to know that more insulin is produced by both human embryonic pancreatic cells and purified Langerhans islets, but as the time advances, there is a gradual decrease of insulin production in graft, but whereas in the purified islets, no change in the insulin production is observed. Some of the researchers cultured the human adult stem cells and modified into transplantable tissue. Usually, beta cells never undergo cell division, but scientists got succeeded in modifying the natural behavior of the beta cells and achieved the cell division of the beta cells. They also made further modifications and made it possible to stimulate the gene which is called as the PDX1 gene. PDX1 gene to produce the insulin. By the culturing of these cells, we can produce normal insulin levels in the diabetic patients. And coming to the next kind of application, the role of stem cells in the treatment of PD, that is the Parkinson's disease. Actually, the Parkinson's disease is a neurodegenerative disease of brain. There is a decrease of control over muscle contraction of the patient. Due to this, during resting period even, involuntary movements of the hands, legs and head takes place. Sustained contraction and loss of balance are also likely to occur. In severe condition, it is highly impossible to speak, walk, and even they are unable to do small things. In some of the Parkinson disease patients, PD patients, the severity of the disease enhanced and that results in the imbalance of their body. They fall down sometimes and the body may get injured, and some of the times these situations lead to the death of the patient. Worldwide, 5 million of people are susceptible for this disease every year per annum. 1% of people at the age of 60, 1% of the people at the age of 60 and 4% of the people at the age of 80. 1% of the people at the age of 60 and 4% of the people at the age of 80, they are susceptible for this kind of Parkinson's disease. A lack of secretion of dopamine. Dopamine is a neurotransmitter that is the main reason for the Parkinson's disease. These specialized neurons of the substantia nigra, this is a region of the brain. These specialized neurons of substantia nigra of the brain, they secrete dopamine. This dopamine acts like the messenger in between substantia nigra and corpus striatum of the brain. As a result, controlled movements happen in an appropriate manner. Due to damage or death of the neurons that secrete dopamine is the main reason for the Parkinson's disease. So, to treat this, the neurons that secrete dopamine, that is, the stem cells which can make the neurons which can secrete dopamine, they are derived from the mice embryonic stem cells. Human embryonic stems are also utilized. Then they are implanted and they differentiate into the new nerve cells. They can produce the dopamine, and this dopamine acts like a neurotransmitter and transmits the nerve signal. So, to make it happen, all these neurons should be substituted in the brain and they have to mingle with the other neurons to work out all this process. Here, the damaged nerve cell failed to make dopamine, and here we will infuse the stem cells. These nerve cells produce the dopamine and the transmission of signal takes place and they regain control over the body of their own. These are the two best examples how the stem cells are utilized in the treating of diabetes mellitus and as well as the Parkinson's disease. Thank you one and all.